Okay, so thank you everyone for coming to this week's Amy seminar. This week is Kareem Wahid, and he's a postdoc at MD Anderson. He's actually a good friend of mine. We went to undergrad together, and we ended up in very similar paths in our postdoc positions. So he'll be talking about the data challenge that he was a part of earlier this year. And go ahead, Kareem, take it away. Thank you.
Sure. Thanks for the introduction, George, and thank you for inviting me today. Share my screen here and make this full screen. All right. So yeah, thanks again. Yeah, I'll be talking about some insights and perspectives from a auto-segmentation data challenge that our group hosted. Yeah, my name's Kareem Wahid, current PhD postdoc at MD Anderson. I have no conflicts of interest. My current funding is the IGCT T32 fellowship here at MD Anderson. This is an overview for the presentation today. I'll present a few quick slides on myself, talk about some general background, and then dive into some results from our data challenge. So yeah, before we get started, I just wanted to give some background on myself. I did my undergrad at UT Rio Grande Valley with George, where I worked on material science research related to radiation detectors, related to radiation detectors. And then I enrolled in a MD PhD program at UT Health, where I worked on machine learning applied to medical imaging, ultimately with the goal of improving the clinical decision-making process for physicians. I then received my PhD in quantitative sciences in 2023, and then decided to pursue a 2-year image-guided cancer therapy fellowship at MD Anderson before finishing my last year of medical school. So fast forward some more to the future, my plans are to pursue a career in radiation oncology and ultimately be able to give back to my community in some capacity as a clinician scientist. And so broadly, the aims of my dissertation work were under the guidance of my mentor, Dr. Clifton Fuller, and were with the goal of using advanced multi-parametric MRI and AI algorithms to develop tools that would help facilitate the detection of head and neck cancer and determining whether those tumors respond to radiation therapy. And for those interested, my dissertation, along with my defense presentation, is publicly available on Figshare. I'll briefly take a few minutes to talk about the T32 postdoc program that I'm currently in. So during my PhD, I focused on developing AI tools for automated image segmentation. What I really wanted to focus at least part of my postdoc on was investigating the translational elements of how you can get tools like that into the clinic. And a lot of that broadly has to do with this idea of human-machine interaction. And so I'm very grateful to have a multidisciplinary mentoring team with a strong research background for this program. So being able to interface with different aspects of research expertise, from imaging physics to biomedical ethics, has been very integral to shaping my own unique research path. Also quickly talk about some T32 activities and opportunities that I've had the chance to participate in. So as part of the program, we trainees participate in a clinical challenges seminar course. And during these, we had the opportunity to have one-on-one discussions with clinicians and researchers on unmet needs in medicine, where emerging imaging technologies can play a role. And as a group, we then put together an AIM sheet, kind of in the style of an R01 or R21, and then gave a presentation on this topic. And some examples are shown on this slide here for the different anatomical sites we did last year. And so I found this exercise immensely helpful, not only to get experience with thinking about science, but also working collaboratively with my peers, which one day may be potentially future collaborators. As part of the program, we also have extensive clinical shadowing opportunities. These are some that I participated in. Actually, today I was spending some time in the nuclear medicine department, which is why I'm wearing scrubs here. And I'll mention a quick anecdote about my time in neurosurgery. I had the wonderful opportunity to shadow Dr. Jeff Weinberg and his team for the first intraoperative MRI case at MD Anderson for thermal ablation for brain metastases. So observing firsthand the complexities of this very advanced imaging technology was very fascinating to me, and an experience that I'm sure I'm going to take with me further on into my career. Grantsmanship is also crucial for this program and a big component of kind of what they want us to get expertise in. So the program has provided valuable resources such as K-Award grant writing workshops. And we also gain hands-on writing experience, with the facilitation of the directors. So I'm not personally submitting something like a K Award for a while, but I'm using these opportunities to generate some aims for other smaller grants, like something called the RSNA Medical Student Research Grant. And one of the aspects of the T32 that I really have enjoyed is, it kind of buys me time to participate in academic activities that I otherwise would not have had, especially if I'd gone directly to medical residency. So, for example, I've had roles on the trainee editorial board for radiology AI, and as an editorial board member for physics and imaging and radiation oncology. These experiences have certainly enhanced my academic and professional development. And just quickly to kind of just show examples of some things that I've done as a trainee editorial board member. I've had the opportunity to do a lot of cool things like host social media chats on topics like cancer imaging, write blog posts on topics like algorithmic fairness. Also had the opportunity to be interviewed for a podcast for some of these educational efforts, and wrote a commentary piece on a big RSNA paper. And yeah, for a lot of these efforts, I'm very fortunate to have received some awards from RSNA, which has also been very nice. And it's kind of a cap off to all this. As of a few weeks ago, I was very fortunate to have been asked to give my first invited talk at a international radiation oncology conference called ESTRO, which is kind of like the big European conference for us. And yeah, I view this as a significant milestone for my career, reflecting the academic productivity and extracurricular involvement that, the T32 has directly supported. And just to kind of summarize, I'm really grateful for the T32 program for, a variety of reasons, but not limited to the multidisciplinary mentorship, clinical translational insights, grantsmanship skills, and academic career preparation. And I'll also say the program is, actively looking for postdoctoral fellows. They have funding for, I think, the next 5 years. So, if any grad students are getting near to the end of their, PhD and are maybe looking for a postdoc opportunity, this is definitely one I would recommend. And I'm happy to talk to any students interested in learning more. Okay, so now I'll move on to more of the research science part of the talk. So I'll discuss some general background first. People in this audience, are likely already familiar with this, but I wanted to give a quick primer on radiotherapy. So the first thing we do is get a few initial images from the patient. Then a radiation oncologist will contour structures on the image that are important, like healthy tissues and the tumor. And using the structures, the planning team will then try to optimize the dose to the tumor. And then finally, the patient will actually have the radiation delivered on the treatment machine. Now, the second part, contouring, which from now on I'm going to refer to as segmentation, has been described as the largest source of uncertainty in radiotherapy. Why? Well, the act of getting humans to segment something on an image is highly variable. This is an example of a head and neck case where 3 physicians were asked to draw out where the tumor is. And this is an issue because considerable inter-observer variability leads to inconsistent segmentation quality, which may be correlated with worse outcomes for patients. And for anyone interested, we've pretty comprehensively characterized human segmentation variability using a large number of observers in a variety of disease sites. Spoiler alert, segmentation variability is often quite high, no matter what structure you're dealing with. Anyways, most people nowadays think the solution to this is artificial intelligence, more specifically deep learning, using well-curated, high-quality data sets. And so deep learning-based auto-segmentation has taken off as a very hot research topic because it has the potential to generate segmentations faster and more consistently than a human. The good news is because of increasing open science practices, the public has access to very standardized and effective deep learning architectures to generate these auto-segmentation solutions. So back in 2015, most of you probably know this, but a very famous architecture called UNet was first developed. And eventually through standard computational libraries, UNet along with its variants have unmistakably become a de facto standard for many medical image segmentation problems. And our group has successfully implemented UNet frameworks for various segmentation-related tasks in radiotherapy, ranging from relatively healthy tissue segmentation to more esoteric tumor-related segmentation. So in addition to standardized approaches, something that has really allowed the field of automated segmentation to flourish is publicly available large-scale data sets. And we briefly kind of talk about this in this review paper that I'm showing here from a few years ago. But public platforms like the Cancer Imaging Archive have really allowed for medical imaging data sets to become accessible to a wider audience. And the radiation oncology applications, that's no exception. So in a similar vein, over the past several years, medical image data challenges, which are public competitions for solving medical image problems, have become powerful tools for democratizing access to large-scale medical data sets and driving community innovation. Here's some examples on the right here from previous head and neck cancer segmentation challenges, which are going to be relevant as we transition to talking about head and neck cancer segmentation in a bit. Um, so I think, probably here in the audience, there's some graduate students, just beginning their graduate studies. And I do want to take a second to vouch for the utility of participating in side projects during your thesis work. And these data science challenges provide a very nice avenue to, work on these side projects in a very standardized and structured way. So it's kind of my own background with data challenges. I was fortunate enough during grad school to participate in a public data challenge called the Hector Challenge, which was head and neck tumor segmentation and outcome prediction in PET CT images. So in 2021, our team entered this competition and was very lucky to place well in some of the various tasks that make up this challenge. And in fact, we actually got first place in a progression-free survival prediction task. And the basic idea with this was that we used a deep learning approach coupled to PET CT data to predict prognostic outcomes for head and neck cancer patients. And ultimately, it was really nice because this got integrated into part of my PhD dissertation, which is pretty cool. This is a screenshot of when we won the competition back in 2021. And because of our success, we were actually invited to participate in the next year's challenge. There's the next year's iteration of the challenge as co-organizers and contributed data sets from MD Anderson for the 2022 edition. So yeah, this is, if you're a grad student, I would highly encourage getting involved with data challenges because you never know what could come out of it. Anyways, based on some of these previously discussed premises, we recently published an editorial that distills some insights for researchers interested in deep learning auto-segmentation for radiotherapy. And I want to briefly focus on this third insight here, which is the idea that in a general sense, auto-segmentation quantitative performance is saturating. Before I go any further, I wanted to give some quick background on how we typically quantify auto-segmentation performance. We often use a geometric measure of segmentation overlap called the dissimilarity coefficient. Basically, if you have two segmentations, you can apply this formula and you get a sense of how well the volumes overlap. And I'm not going to go into in this presentation, but this metric has a lot of flaws, especially for radiotherapy, but it's currently the standard way that we often evaluate auto-segmentation quality. So back to this idea of performance saturation. Over the past several years, medical image data challenges have been inundated with these variants of the UNet architecture that I previously mentioned. And this surge has seemingly decreased the gap between state-of-the-art and average participant performance. And so for in radiotherapy-related segmentation, the Hector challenge actually stands out as a prime example of this. This graph shows that over time, state-of-the-art performance, which are those red dots, has steadily plateaued. And moreover, once a measure of human performance benchmarking has been exceeded, which in this case is inter-observer variability represented by this gray line, the practical benefits of further improving geometric indices becomes somewhat ambiguous. So kind of with all that being said, it may seem like auto-segmentation using deep learning tools is a mostly solved problem for many applications. So what else should the research community be focusing on? Well, there's actually a lot, but there's limited time for this presentation. So I'll just focus on two quick things, and that is uncertainty quantification and investigating novel application spaces. And so what do we mean when I say prediction uncertainty or uncertainty quantification? Broadly, this means we want to return a distribution over these predictions rather than a single point estimate. And in a classic classification framework, which is more relevant for our segmentation kind of discussion, this can look like outputting a confidence score along with the output label. And generally, good uncertainty estimates will quantify when you can trust that model's predictions. So we recently conducted a scoping review of AI uncertainty quantification in radiotherapy, and shown kind of as expected, people are mainly applying these techniques right now for auto-segmentation applications as part of the contour QA process. And so there's a lot of research focus on this area right now, which I'm diving into as part of my postdoc, but don't have time to talk about just right now in this presentation. So just a quick plug, I guess, for this review article if you're interested in learning more about this topic. But yeah, for the focus of this presentation, another aspect of important research directions is investigating novel segmentation application spaces or unmet needs where little research has been done previously. And so this brings me to our data science challenge titled Head and Neck Tumor Segmentation for MR-Guided Applications, or HNTS-MRG for short. So yeah, I'll spend the rest of this time briefly talking about this data challenge that we organized from the ground up. To give some quick background on motivation for the challenge, MRI-guided radiotherapy is a cutting-edge technique in head and neck cancer treatment. Due to the ease of acquiring longitudinal imaging, it allows us to potentially target tumors with much greater precision than the conventional way that we do it with CT. And so that's kind of where, however, manually segmenting tumors is exceptionally time consuming. So that's where, AI comes in, and data challenges like Hector, which I previously mentioned, have shown the potential of AI in advancing tumor auto-segmentation. But there's still this gap in knowledge, especially when it comes to large AI-ready data sets for adaptive radiotherapy purposes. So with this in mind, we developed the Huntsmer's 2024 challenge in collaboration with Mackay. Um, and here's a general timeline of events for the challenge. We opened our website in April, released the training data in June, opened the evaluation phases in August, and then had a live wrap-up event in October. And I'll take a quick second just to plug the grandchallenge.org site, which is where we hosted this challenge out of. It's a very cool site that provides a platform for end-to-end development of machine learning solutions in biomedical imaging. You can host data challenges like we did, upload algorithms, and host, clinician reader studies, amongst other things. I will, however, say, hosting a challenge is a bit pricey. So there's a base cost of 5,000 euros right now, plus extra depending on challenge specifications. Um, these are just some screenshots from our Grand Challenge website, which featured several descriptive pages and a forum for real-time discussion. And just to kind of quickly show you, or for you to get a sense of how the Grand Challenge works for hosting data challenges, the basic workflow is participants will submit a Docker container image containing their algorithm solution. And then that image gets applied to each individual case or image in your data in the test data set. And you get an individual output, in this case, a segmentation output for every single item in that archive. And then us as challenge admins have a separate Docker container image that evaluates segmentation outputs. And so that gets concatenated with, the output of the participants' algorithms. And that allows us to get a kind of like a JSON of all the metrics that we want to calculate. And then that gets displayed on the website. And so like Grand Challenge has kind of like done all the heavy lifting of the back end stuff for us. So we really just have to like tweak a few things, make a few Docker containers, and then the whole thing is good to go basically. Um, in order to more easily interface with all this minutia on the Grand Challenge site, we also provided users with a GitHub containing various resources for algorithm development. And we also generated some video tutorials for various aspects of the challenge, which can be found on the Huntsmer's YouTube page. So as far as the data source, we focused on T2 weighted MRI scans of the head and neck, primarily involving patients with oropharyngeal squamous cell carcinoma treated with radiotherapy. Overall, our data set included 202 patients. And from these patients, we collected images at two key time points, pre-radiotherapy and mid-radiotherapy. And these scans were acquired using a variety of MRI scanners, ensuring that we captured a broad range of imaging protocols. And here's an example of some imaging data from two patients. The patient in the top row had a non-fat suppressed T280 sequence, while the patient in the bottom row had a fat suppressed sequence. So this shows the potential diversity of images you can see in the data set, even though we're all technically T2 weighted. And in this study, we focused on two critical structures for segmentation targets, and that was the gross primary tumor volume, or GTVP, and metastatic regional lymph nodes, or GTVN. And these are kind of like the bread and butter of what the radiation oncologist is going to be segmenting in the clinic. So each case was independently segmented by 3 to 4 expert physician annotators. And in total, we had 13 unique physician annotators contribute to this data set, each with at least 2 years of experience in the field. And these physicians had access to patient medical histories and prior imaging to aid in their segmentations if they needed. To combine these multiple annotations into a single reference standard, we use the simultaneous truth and performance level estimation algorithm, or staple algorithm, which is a mostly widely accepted method for generating consensus segmentations from multiple inputs. And these segmentations were all checked by a senior radiation oncology faculty member. And in cases where there were significant disagreements between observers, like disputes over the extent of lymph node involvement, which is pretty common, the final decision was deferred to a senior faculty member who generated the final segmentation. Um, this is a visual example of the mask labeling scheme for this challenge. So here, the green is the GCP or primary tumor, and the yellow is the lymph node. Regarding data processing, we converted the DICOM images, which are the raw format data, into and structure files into NIfTY format. And additionally, we cropped the images to focus specifically on the head and neck region. So this ensured greater consistency across the data set and also removed facial features without affecting the underlying segmentation masks. And as an additional data pre-processing, we registered the pre-RT images to mid-RT images. We utilized a registration framework using SimpleITK and elastics for deformable registration. Specifically, we used a parameter map from the elastics model zoo that had been validated in previous studies using very similar data. Yeah, so from the 202 patients we collected, we split this into 150 cases for training and held that 52 cases for private evaluation. And we partitioned the data to ensure similar distribution across key factors like image fat suppression status, tumor response, staging, HUV status, etc. And that equal partitioning is kind of illustrated in these plots here. We publicly released the training data set of images and masks on Zenodo under a Creative Commons license, which allowed users to quickly get access to the data. And as far as tasks, this challenge was split into two separate tasks, and participants could participate in one or both of the tasks. So task one focused on predicting segmentations on unseen pre-RT images, as this task is analogous to previous tumor segmentation challenges like Hector. And task two focused on predicting segmentations on unseen mid-RT images. And kind of by definition, this was a more complex task since participants could use multiple inputs, including both the pre-RT and mid-RT images, along with the registered data to develop their algorithms. And to date, there hadn't been any challenges that utilized patient-specific prior data for model development, making task two particularly unique. So for this challenge, we employed the aggregated dice similarity coefficient, or dice-ag, to evaluate the segmentation results. I won't go into details, but this metric is very similar to the standardized coefficient that I talked about earlier, but can better account for scenarios where some structures may not be present in a particular image. And so we calculated the aggregated dice for both GTVP and GTVN separately, and then averaged the two for the final ranking of the challenge. And so, like I mentioned at the beginning, the submissions for this challenge were managed using Docker containers to ensure a standardized and fair evaluation process. And so the submission process was divided into two phases. The first was the preliminary development phase, which was designed to help participants familiarize themselves with Docker. And the second phase was the final test phase, where participants submitted their final algorithm for evaluation. This involved evaluating the algorithms on 50 unseen test cases, and they could only submit once for that. We had some baseline models for the challenge, kind of as benchmarks. And so we used the NN unit architecture, which is kind of the industry standard for building baseline models. For task one, we trained models using only pre-RT images. And in task two, the models were trained similarly, but this time with mid-RT images as input. We also included a null model for task two as a simple baseline. And this model essentially just used the registered pre-RT segmentations directly as mid-RT predictions, kind of just as a simplified approach. And I'm happy to report that we had a pretty strong participation in this challenge with 107 registrations from around the globe. And ultimately, after all was said and done, 19 teams were determined to be eligible for the leaderboards. I'd also like to quickly talk about inter-observer variability in our data set. Since we had multiple annotators per case, we could actually calculate measures of inter-observer variability, which could also serve as benchmarks for the challenge. So we calculated both traditional dice and aggregated dice to quantify the consistency between annotators across multiple cases. And so the traditional dice offers this pairwise geometric comparison between observers, which allows for a straightforward measure of variability. And this is kind of what you see in a lot of papers when they mention inter-observer variability. Um, the box plot here shows the values for that. Um, but more germane to our challenge, we also calculated a measure of variability for aggregated dice. Um, and so calculating this was a little more involved, but to briefly summarize, we first computed the pairwise intermediate measures between every annotator's segmentation for each case. And these pairwise values were then aggregated across all cases for each unique annotator pair. So you end up with an aggregated dice for observer 1 versus observer 2, and then another aggregated dice for observer 1 versus observer 3, etc. And so lastly, we derived the final inter-observer variability aggregated dice score by taking a weighted average of all these annotator pair values. And the weight is determined by the actual number of cases that each annotator pair contributes. Um, and those numbers are shown here, which are fairly close to the measures of that we calculated with traditional dice, but are slightly higher, which indicate, I guess, less variability. Um, and we'll talk more about the specific numbers for inter variability in the next few slides. But just to quickly mention, for pre-RT aggregated dice, we got a mean value of 0.806. And for mid-RT, we got a mean value of 0.714. And the inter variability got worse, particularly for the mid-RT GTVP structure, which decreased from 0.757 to zero point 0.6. Which is mostly expected since mid-RT segmentation is a harder process for clinicians than pre-RT segmentation. And also quickly note that we excluded cases in these calculations where only the senior faculty annotator provided the final segmentation, due to the significant observer disagreement, which is actually might be setting a slightly higher bar than usual for these measures. Um, okay, so now I'll go ahead and talk about results from the participant algorithms for the challenge. And we'll start with the task one results. So we saw an impressive range of aggregated dice results from our 18 participating teams, with mean scores ranging from 0.571 to 0.825, and an overall mean across all the teams of 0.783. And here's some additional comparators, including the NNUNet baseline and inter-observer variability, which we measured from the clinician annotators. So the top 50% of participants, or the top 9 teams here, outperformed the inter-observer variability threshold of 0.806, showing their strong performance. And here's a box plot summary of all the results for task one, which shows the aggregated dice mean, the GTVP, and GTVN scores. To quickly summarize all that with some text, task one demonstrated high performance overall, with the top teams achieving mean aggregated dice results around 0.8. These results align closely with previous tumor segmentation challenges like Hector 2022. And as mentioned, the top 50% of teams performed as well as or better than inter-observer variability, highlighting the strength of these participants' approaches. And one of the main observations was that the GTVP structures were consistently more challenging to segment than GTVN for the algorithms. And finally, one observation we noticed for task one was that NNUNet-based solutions predominated for the participant approaches. In fact, our baseline NNUNet model performed fairly strongly, achieving 4th place in the overall ranking. Okay, now to delve into general results for task two. Here we had 15 teams, and the mean aggregated dice results range from 0.562 to 0.733, with an overall mean of 0.688. To show some additional comparators, we have here the NNUNet baseline, the null algorithm, and inter-observer variability. And most teams perform better than the NNUNet baseline and the null algorithm. But interestingly, only the top 4 teams, which make up the top 25% of participants, managed to exceed inter-observer variability in these results. So here's a box plot summary of those results. To quickly summarize this in text, task 2 proved to be more challenging than task 1, with only the top 25% of teams surpassing inter-observer variability. And one of the key challenges was the segmentation of GTVP structures, likely due to tumor shrinkage and radiation-induced effects complicating the contouring process for mid therapy. And compared to GTVP, the GTVN segmentations were relatively easier in task 2, so even more so than in task 1. And this is likely because large nodes kind of persisted often at mid therapy, making the segmentation process simpler, especially when you're incorporating patient-specific prior pre-RT data. Interestingly, the baseline NNUNet underperformed when compared to inter-observer variability, with even a simple null model achieving similar results. However, most submitted algorithms outperform both the NMUNet baseline and the null model, indicating the utility of these approaches. And ultimately, the most successful methods demonstrate the value of integrating registered patient-specific data into the model development, and this clearly added some kind of advantage for this more complex task. Um, to kind of just holistically get a sense of the results across the two tasks, here's a box plot showing the combined results. And you can really appreciate here the big difference in performance between task one and task two, especially for that GTVP structure. Um, Finally, to assess whether there was any relationship between performance in both tasks, we conducted a correlation analysis for participants who completed both of the tasks. And interestingly, the correlations were generally very weak, suggesting that strong performance in one task didn't necessarily predict strong performance in the other. And so obviously I don't have time to go into details about the specific participant methods, but as part of the challenge, the participants were required to submit a short paper describing their methods and results. And these papers have just finished peer review actually, and are going to be included in the Springer Lecture Note in Computer Science Proceedings, which we're hoping to have published in the next few months. Also, to wrap things up for the competition, we had a live virtual event that we held at the end of October, where the top teams were invited to present their methods, and the official winners were announced. These are some screenshots from the event that day, and we actually recorded this, so we'll have this uploaded on YouTube in the next few months. Okay, and to just kind of close out here, I thought it could be a fun little exercise to plot my stress related to organizing all this as a function of time. So we start off with very manageable levels, but they quickly rise as I begin to realize that our physician annotators are waiting until the last minute to complete their segmentations. We get past the test date release, and this little spike here is from me thinking I accidentally publicly pushed some test data onto our GitHub, but luckily I did not. Stress begins to increase as I get inundated with emails from participants who decided to wait till the last minute to submit their Docker containers. So I'm stuck, being, stuck with very late nights debugging people's Docker containers. This little peek here is us actually running out of compute budget on the grandchallenge.org site, literally at the last hour of the submission deadline. So me having to deal with all that, that was kind of crazy. And then things die down for a little bit, but naturally get more stressful as we get closer to our live event. As I realize our presenters are all waiting till the last minute to send me their presentations. So there's a recurring pattern here that you might be noticing. Then finally, our next nearest deadline was for the peer review process, and naturally people waited till the last minute to send me the reviews. So it looks a little bit like an EKG if you squint hard enough, and that's probably appropriate here. And so one thing I can say, with all this is, patience is a virtue. A lot of things are out of your control. As long as you're doing your best and trying to move things along, things will fall into place. There's easier said than done, but no need to stress yourself out over things that are ultimately out of your control, right? Anyways, I hope you can appreciate that, this was a stressful process, but to me, it all, kind of becomes worth it when I see messages like this in my inbox. So this is a message from one of the participants saying, you guys are all awesome, definitely know how to run challenge effectively. And these here, this person says, I can't repeat enough how well organized all your efforts are. This person's very nice and saying, you're the most responsible challenge organizers I've seen. And here's one of my favorites. If you come to Shanghai one day, you can contact me. I will take you to taste the food. So I may end up taking that person up on their offer. So yeah, at the end of the day, I think it was a stressful process, but ultimately worth it in my mind. Okay, now while I'm getting to the end here, I'll discuss some limitations. The challenge provided some valuable insights, but naturally there's some limitations to acknowledge. First, the data set was fairly modest in size and came from a single institution, which may limit its generalizability. It also focused specifically on oropharyngeal tumors instead of head and neck cancer more generally. Importantly, we relied on typical clinician annotation workflows. But if we had, strict annotation guidelines, that would have been very beneficial to ensuring more consistency in our data set. Moreover, there was potential for expanding the data set with additional imaging modalities like PET-CT or multi-parametric MRI. In fact, we really wanted to incorporate diffusion weighted imaging into this challenge, but we ran into some issues with, you know, us like having to lose too many patients if we wanted to do that. So that was a whole other thing. Um, Also, we aim for a robust evaluation using aggregated dice, but our choice of evaluation metrics could have been expanded and probably should be in future iterations. Um, and finally, the Grand Challenge backend actually didn't have the best GPUs that I had access to. So when people submitted Docker containers, sometimes you would run out of like time for inference. We had like a 20-minute limit, and like they were used to like these very good GPUs running things very quickly, but on the Grand Challenge, it wasn't running that quickly, and they had to like modify their approaches. So, the last time I checked, the Grand Challenge is trying to upgrade, the cloud-based GPUs that they're using, but that was a limitation here. Um, and we do have plans for a comprehensive public release of the dataset through the Cancer Imaging Archive. The training data is already available on Zenodo, but we want to put out a more comprehensive version of the data. And so this release will include the full training and test data sets, so 202 cases in total. The raw DICOM files and individual annotator segmentations. And these individual segmentations would hopefully enable researchers to perform uncertainty quantification experiments like I was talking about earlier, such as ambiguity modeling. Um, Additionally, we would provide clinical metadata to enrich the data set as well. And to ensure the ease of use for the research community, we're also writing a data descriptor that will be published alongside the release of this entire data set. We're also excited to announce plans for a post-challenge secondary analysis paper, which we're hoping to submit to a high impact field-specific journal. So this paper will conduct a kind of a meta-analysis of the algorithms that were submitted, exploring topics like inter-algorithm variability, consensus segmentation experiments, and patient-specific sub-analyses. And this paper would be in line with previous secondary analysis papers, like the Hector Challenge did, published recently in Medical Image Analysis. And eligible participants who successfully submitted their Docker containers and papers will be invited to be co-authors on this manuscript. So at this point, there's no concrete plans for a second edition of the Huntsmerge Challenge, but future iterations definitely remain a possibility. If we do move forward, future editions could include more diverse imaging sequences and additional intra-treatment images, potentially with a focus on MR-Linac technology. Additionally, we would aim to include multi-institutional data to increase the dataset size and diversity, capturing a wider range of imaging protocols and patient populations. So expanding the data set in that way would provide broader insights into the performance of MR-guided radiotherapy segmentation algorithms. Okay, so to kind of wrap things up here, the Huntsmerge 2024 challenge successfully tackled MRI-based automated tumor segmentation for pre-RT and mid-RT. The challenge attracted significant global interest with 19 final eligible teams. Task 1 saw strong performance, with most algorithms achieving scores that surpassed clinician inter-observer variability. Task 2, however, proved to be more challenging. The top algorithms did manage to outperform clinician inter-observer variability. And across both tasks, segmenting the primary tumor consistently posed a greater challenge compared to the lymph nodes. And looking ahead, future efforts should focus on harmonizing clinician guidelines, increasing dataset diversity, and expanding the range of evaluation metrics utilized. And yes, ultimately, a stressful process, but worth it, I think. So yeah, these are my acknowledgements. I'd like to thank George for inviting me to speak today. I'd also like to acknowledge the IUCT T32 program, my mentorship team, the Fuller Radiotherapy Lab, the Fuentes Computational Lab, and my various collaborators. And finally, thank you to the audience for your time. Here's my contact info for anyone who wants to reach out to discuss, research stuff, career stuff, or whatever. I'm happy to always talk to trainees or anybody. So yeah, okay. I'm happy to take any questions if there's time, George.
Yeah, thanks, Kareem. Let's give Kareem a round of applause for a very interesting talk on the data challenge. And if you have any questions, um,